Welcome to our comprehensive guide on molecular interactions, a foundational topic in biochemistry. In this lesson, we'll explore the different types of bonds and forces that hold molecules together and drive the chemistry of life. From the strongest covalent bonds to the weakest van der Waals forces, understanding these interactions is key to grasping how biological molecules behave. You'll also see how these forces influence the structure and function of proteins, nucleic acids, and other biomolecules. Let's begin by setting the stage for why these interactions matter in the context of living systems. Covalent bonds are the strongest forces that hold atoms together in a molecule. In a covalent bond, two atoms share one or more pairs of electrons, creating a stable connection. This sharing can be equal or unequal, depending on the atoms involved. Covalent bonds are fundamental in forming the backbone of organic molecules like proteins, DNA, and carbohydrates. Their strength ensures that the basic structure of these molecules remains intact, even under various physiological conditions. Next, we'll look at the difference between polar and nonpolar covalent bonds. Not all covalent bonds are created equal. When two atoms with different electronegativity share electrons, the bond is polar covalent. This means electrons are shared unequally, creating partial positive and negative charges within the molecule. Water is a classic example of a polar covalent molecule. In contrast, nonpolar covalent bonds occur when atoms share electrons equally, as seen in molecules like methane. The polarity of these bonds has a profound effect on molecular behavior, influencing solubility, reactivity, and interaction with other molecules. Electronegativity is a measure of how strongly an atom attracts electrons in a bond. The greater the difference in electronegativity between two atoms, the more polar the bond. For example, fluorine is highly electronegative, while hydrogen is much less so. When these two form a bond, the electrons are drawn closer to fluorine, creating a significant dipole. Understanding electronegativity helps predict the behavior of molecules in different environments, including how they interact with water and other solvents. Let's compare three important molecules: water, methane, and carbon dioxide. Water has polar covalent bonds and a bent shape, making it a polar molecule. Methane, with nonpolar covalent bonds and a tetrahedral shape, is nonpolar. Carbon dioxide has polar covalent bonds, but its linear shape causes the dipoles to cancel out, resulting in a nonpolar molecule. These differences in bond polarity and molecular shape explain why water is an excellent solvent, while methane and carbon dioxide behave differently in biological systems. Molecular shape plays a crucial role in determining a molecule's properties. For example, the bent shape of water leads to its polarity, while the linear shape of carbon dioxide results in a nonpolar molecule. Visualizing these shapes helps us understand how molecules interact with each other and with their environment. The three-dimensional arrangement of atoms affects everything from boiling point to biological function. Next, we'll explore how unequal sharing of electrons leads to partial charges and molecular dipoles. When electrons are shared unequally in a bond, partial charges develop on the atoms involved. This creates a dipole, with one end of the molecule slightly positive and the other slightly negative. These dipoles are responsible for many of the unique properties of molecules like water, including its ability to dissolve a wide range of substances. The presence of dipoles also enables molecules to interact through dipole-dipole attractions, which we'll discuss in more detail later. Strength and length of a bond are closely related. Generally, the stronger the bond, the shorter its length. Triple bonds are stronger and shorter than double bonds, which are in turn stronger and shorter than single bonds. This relationship affects the stability and reactivity of molecules. For example, the triple bond in nitrogen gas makes it very stable, while single bonds are more easily broken and reformed during chemical reactions. Atoms can exist in different energy states. In the ground state, electrons occupy the lowest available energy levels. When energy is absorbed, electrons can move to higher excited states. However, bonding typically involves electrons in the ground state, as these are more stable. Excited states are important in processes like photosynthesis and fluorescence, where energy transfer plays a key role. Understanding these states helps explain why certain bonds form and how molecules absorb and emit energy. Different types of bonds have different energies. Covalent bonds are the strongest, requiring the most energy to break. Ionic bonds are also strong, but generally weaker than covalent bonds. Metallic bonds, hydrogen bonds, and van der Waals forces are progressively weaker. This hierarchy of bond strengths is crucial in biology, where weak interactions allow for dynamic processes like enzyme activity and DNA replication, while strong bonds provide structural stability. While strong bonds hold molecules together, weak interactions play a vital role in the structure and function of biological macromolecules. These include hydrogen bonds, dipole-dipole interactions, van der Waals forces, and hydrophobic interactions. Although individually weak, collectively they stabilize complex structures like proteins and DNA. Their transient nature allows for flexibility and dynamic changes, essential for biological processes such as enzyme catalysis and molecular recognition. Hydrogen bonds are a special type of weak interaction that occurs when a hydrogen atom covalently bonded to a highly electronegative atom, like oxygen or nitrogen, is attracted to another electronegative atom. These bonds are much weaker than covalent bonds, but are crucial in stabilizing the structures of proteins and nucleic acids. In water, hydrogen bonds are responsible for its high boiling point and surface tension. Let's look at how hydrogen bonds form and why they matter. Energy required to break a hydrogen bond is much less than that for a covalent bond. For example, in water, a hydrogen bond has a bond dissociation energy of about 23 kJ/mol, compared to 470 kJ/mol for the covalent OH bond. This difference in energy explains why hydrogen bonds are easily formed and broken, allowing for the dynamic nature of biological systems. Hydrogen bonds are especially important in the secondary structure of proteins and the base pairing in DNA. Hydrogen bonds are found throughout biological molecules. In DNA, they hold together the complementary base pairs, ensuring the stability of the double helix. In proteins, hydrogen bonds stabilize secondary structures like alpha helices and beta sheets. These interactions are also crucial in the formation of water networks and the folding of amino acid side chains. The versatility and specificity of hydrogen bonds make them essential for the proper functioning of living organisms. Water is often called the universal solvent, and its ability to form hydrogen bonds is a major reason why. Each water molecule can form up to 4 hydrogen bonds with neighboring molecules, creating a dynamic network. This network is responsible for water's high boiling point, surface tension, and its ability to dissolve many substances. The polarity of water molecules and their hydrogen bonding capacity make water an ideal environment for biochemical reactions. Only atoms with high electronegativity, such as fluorine, oxygen, and nitrogen, can participate in hydrogen bonding. The large difference in electronegativity between hydrogen and these atoms creates a strong dipole, enabling the formation of hydrogen bonds. This is why hydrogen bonds are commonly found in water, ammonia, and organic molecules containing, or NH groups. The strength and directionality of hydrogen bonds are crucial for the three-dimensional structure of biomolecules. Hydrogen bonds are strongest when the 3 atoms involved, donor, hydrogen, and acceptor, are aligned in a straight line. This geometry allows for maximum overlap of orbitals, resulting in a stronger interaction. In biological systems, this alignment is often achieved in the secondary structures of proteins and in the base pairing of nucleic acids. The directionality of hydrogen bonds contributes to the specificity and stability of molecular interactions. Strength of a hydrogen bond depends on the bond angle and the distance between the atoms. The optimal bond angle is close to 180 degrees, which allows for the strongest interaction. As the angle deviates from this ideal, the bond becomes weaker. Similarly, the shorter the distance between the donor and acceptor atoms, the stronger the hydrogen bond. These factors are important in determining the stability of protein and nucleic acid structures. Hydrazine and glycerol are 2 molecules that can form multiple hydrogen bonds due to the presence of several NH or O groups. Hydrazine, with its 2 NH2 groups, can form up to 4 hydrogen bonds, making it highly soluble in water. Glycerol, with 3 O groups, also forms extensive hydrogen bonding networks. The ability to form multiple hydrogen bonds increases a molecule's solubility and affects its physical properties, such as boiling point and viscosity. Hydration power of a molecule depends on the number of hydrogen bonds it can form with water. Hydrazine, glycerol, and water all have high hydration power due to their ability to form extensive hydrogen bonding networks. This property is important in biological systems, where hydration shells around ions and molecules influence their behavior and interactions. Hydration power also affects the solubility and transport of molecules in aqueous environments. Van der Waals forces are the weakest of all molecular interactions. They arise from transient dipoles generated by the rapid movement of electrons in atoms and molecules. These forces act over very short distances and decrease rapidly as the distance between atoms increases. Despite their weakness, Van der Waals forces are important in stabilizing the structures of large biomolecules and in mediating interactions between nonpolar molecules. Van der Waals interactions include attractions between 2 pre-existing dipoles, between a dipole and an induced dipole, and between 2 induced dipoles. The last type, known as London dispersion forces, is the only attractive force between nonpolar molecules. These interactions are always present, regardless of the charges or electronegativities of the atoms involved. Their cumulative effect can be significant, especially in large molecules or assemblies. London dispersion forces are a type of Van der Waals interaction that arises from temporary imbalances in electron distribution. These transient dipoles induce similar dipoles in neighboring molecules, leading to a weak attraction. Although individually weak, London forces become significant in large molecules or when many such interactions occur simultaneously. They play a key role in the condensation of nonpolar gases and the stabilization of lipid bilayers. Dipole-dipole interactions occur when the partial charges on polar molecules attract each other. The positive end of one molecule aligns with the negative end of another, creating a stabilizing force. These interactions are stronger than Van der Waals forces, but weaker than hydrogen bonds. Dipole-dipole interactions are important in determining the boiling and melting points of polar compounds and in the alignment of molecules in biological membranes. Hydrophobic interactions refer to the tendency of nonpolar molecules or groups to associate with each other in an aqueous environment. This self-association minimizes the disruption of hydrogen bonding among water molecules, leading to the aggregation of nonpolar substances. Hydrophobic interactions are crucial in the folding of proteins, the formation of cell membranes, and the assembly of lipid bilayers. They are driven by the exclusion of water, rather than by direct attraction between nonpolar groups. Hydrophilic molecules dissolve readily in water due to their ability to form hydrogen bonds or ionic interactions with water molecules. These substances are composed of ions or polar molecules that attract water through electrical charge effects. Water molecules surround each ion or polar molecule, carrying it into solution. This property is essential for the transport of nutrients, waste, and signaling molecules in biological systems. Ionic substances like sodium chloride dissolve in water because water molecules are attracted to the positive and negative charges of the ions. Polar substances, such as urea, dissolve by forming hydrogen bonds with water. The ability of water to dissolve both ionic and polar compounds makes it an excellent solvent for a wide range of biological molecules, facilitating chemical reactions and transport within cells. Molecules with many nonpolar bonds, such as hydrocarbons, are usually insoluble in water and are termed hydrophobic. Water molecules are not attracted to these substances and do not surround them, leading to their exclusion from the aqueous environment. This property is important in the formation of cell membranes and the compartmentalization of biological processes. Hydrophobic effect drives the folding of proteins by causing nonpolar side chains to cluster in the interior of the molecule, away from water. This minimizes the disruption of hydrogen bonding among water molecules and stabilizes the protein's three-dimensional structure. The hydrophobic effect is a primary force in the formation of biological membranes and the assembly of macromolecular complexes. Let's summarize the main types of chemical bonds. Covalent bonds involve the sharing of electrons and are the strongest. Ionic bonds result from the transfer of electrons and the attraction between oppositely charged ions. Metallic bonds involve a sea of delocalized electrons. Hydrogen bonds, van der Waals forces, dipole-dipole interactions, and hydrophobic interactions are all weaker, but play crucial roles in the structure and function of biological molecules. Bond energies vary widely, from the strong covalent bonds that hold biomolecules together to the weak van der Waals forces that allow for dynamic interactions. In biological systems, the balance between strong and weak interactions is essential. Strong bonds provide stability, while weak interactions allow for flexibility, recognition, and regulation. This balance is at the heart of molecular biology and biochemistry. Water's unique properties stem from its polar nature and its ability to form hydrogen bonds. As the universal solvent, water dissolves a vast array of substances, facilitating chemical reactions and transport in living organisms. Its high specific heat, surface tension, and heat of vaporization are all consequences of hydrogen bonding. Water's role as a solvent is central to life as we know it. Water molecule consists of two hydrogen atoms covalently bonded to an oxygen atom. The electrons are distributed asymmetrically, making water a polar molecule. This polarity allows water molecules to form hydrogen bonds with each other, creating a dynamic, flickering network. The cohesive nature of water is responsible for its unusual properties and its ability to support life. Hydrogen bonds between water molecules are responsible for many of water's unique properties. These bonds are transient, constantly forming and breaking, but at any moment, a significant fraction of water molecules are hydrogen bonded to their neighbors. This network gives water its high surface tension, specific heat, and heat of vaporization, all of which are vital for biological processes. Length of a hydrogen bond is longer than that of a covalent bond. For example, the OH covalent bond in water is about 0.1 nm, while the hydrogen bond is about 0.17 nm. The van der Waals radius defines the closest distance at which two non-bonded atoms can approach each other. These measurements are important in understanding molecular packing and interactions in biological systems. Water's physical properties, such as its high boiling point, surface tension, and ability to dissolve many substances, are direct results of its molecular structure and hydrogen bonding. These properties enable water to support life by maintaining temperature stability, facilitating chemical reactions, and allowing for the transport of nutrients and waste. Solubility of gases in water depends on their polarity. Polar gases like ammonia and hydrogen sulfide are much more soluble than nonpolar gases like nitrogen and oxygen. This trend reflects the ability of water to form hydrogen bonds or dipole interactions with polar molecules, while nonpolar gases interact only through weak van der Waals forces. Weak interactions, such as hydrogen bonds, ionic interactions, van der Waals forces, and hydrophobic effects, are crucial for the structure and function of macromolecules. These forces stabilize the three-dimensional shapes of proteins, nucleic acids, and other large molecules, allowing them to perform their biological roles effectively. The dynamic nature of these interactions enables molecular recognition, signaling, and catalysis. Proteins are stabilized by a combination of strong covalent bonds, such as peptide and disulfide bonds, and weak interactions like hydrogen bonds, ionic interactions, and hydrophobic effects. The interplay between these forces determines the protein's final shape and its ability to function. Disruption of these interactions can lead to denaturation and loss of biological activity. Nucleic acids, such as DNA and RNA, are held together by strong covalent bonds in the backbone and weak hydrogen bonds between complementary bases. The balance between these interactions allows for the stable storage of genetic information and the dynamic processes of replication and transcription. Carbohydrates are linked by glycosidic bonds, while lipids are held together by ester and phosphoester bonds. These covalent bonds provide structural integrity, while weak interactions help organize these molecules into larger assemblies, such as cell walls and membranes. Major classes of biomolecules, proteins, nucleic acids, carbohydrates, and lipids, are built from smaller subunits linked by covalent bonds. These building blocks assemble into larger structures through both strong and weak interactions, enabling the complexity and diversity of life. Macromolecules are formed by condensation reactions, which join subunits and release water. The reverse process, hydrolysis, breaks down polymers into monomers by adding water. Condensation reactions are energetically unfavorable and require energy input, while hydrolysis is energetically favorable. These reactions are central to metabolism and the regulation of cellular processes. Classic example of condensation is the reaction between a carboxylic acid and an amine, forming an amide bond and releasing water. This type of reaction is fundamental in the formation of proteins, where amino acids are linked by peptide bonds. Understanding these reactions helps explain how macromolecules are synthesized and broken down in living organisms. In summary, the interplay of strong and weak molecular interactions underpins the structure, function, and dynamics of all biological molecules. From the stability provided by covalent bonds to the flexibility and specificity enabled by weak forces, these interactions are at the heart of biochemistry. Understanding them is essential for anyone studying life at the molecular level. As you continue your journey in biochemistry, keep these fundamental concepts in mind. They are the key to unlocking the mysteries of life.